All right. Well, thank you all for being here today. I'm Mary Ann Pohl, and I'm a Director of Alliance Management at Ailux Biologics. And today I'm going to talk to you about using AI for antibody discovery and engineering and how we do it at Ailux. So just to give you an overview on our company, we are the Biologics division of XtalPi or XtalPi, which is a company that's been around for more than 10 years now and is partnered with 15 of the 20 top pharmaceutical companies on small molecule drug discovery projects and other programs. But within Ailux, we have over 80 team members across 3 different countries, and we have developed our own proprietary suite of AI models, along with a fully established wet lab space with all of the instrumentation you need for antibody discovery. And currently, we've worked with over 20 strategic partners and have worked on over 60 antibody discovery and engineering programs. So we all know that AI is trending in antibody discovery. It's a hot topic at this conference and others, and you can see that based on the number of publications related to AI and antibody discovery over recent years. And a lot of people have made a lot of claims about what AI can do in our field. And so the question becomes, is AI really having an impact, or is it a lot of hype? And so I hope to address that question today with my talk. I don't want to give you any hype. I want to give you 2 case studies that show how at Ailux we've been able to incorporate AI into our discovery and engineering workflows. So the first case study I'm going to share with you today is related to hit generation, and then I'm going to talk about a next-gen engineering case study. But before I get into the case studies, I want to present to you the 3 overarching AI engines that we've developed at Ailux Biologics. So the first one is CrystalFold, which is our AI-powered complex structure prediction platform. So with this, we can take just primary sequence data of our antibody and our antigen and feed that into the AI platform, and it will give us a complex structure prediction that is highly accurate. So we can use this CrystalFold platform for a whole host of antibody engineering tasks, and I'll show you how later on. Then we have Zenprot, which is our generative AI engine, and we use this for increasing the success rate for expression and overall developability when generating antibody variants compared to just random mutagenesis. And then finally, we have Zentient, which is our predictive suite of AI algorithms, and this is a whole host of algorithms that we've developed for predicting key features like developability and binding, and can help us really triage as we're going through candidate selection. And so the advantage of this platform is it really allows us to pare down the number of antibodies that we have to actually express and validate in the wet lab. And so in my experience, I come from a more traditional antibody discovery background where we're used to producing and characterizing hundreds and hundreds of clones. And with the power of Zentient, we found that we can express and test a much fewer number to still find clones that meet our antibody design goals. So these are the 3 main platforms that we work with at Ailux. So now I'll get into the first case study related to antibody discovery and give you an overview of our Trilux platform. So Trilux is a signature approach that we've developed that allows us to identify diverse, functional, developable, and also humanized hits. And the case I'm going to show you today is against a novel immune checkpoint target. So we all know that the immune repertoire is often underexplored. So when we use more traditional hit identification approaches, of course we're able to identify expanded clonotypes in the repertoire because they're overrepresented in NGS data, or you just have a much higher probability of picking them in a B cell or phage selection. However, within that repertoire, there are many singleton clonotypes that are rare in that NGS data set that may escape examination, but they can be very valuable clones. And I'm going to show you how with the Trilux platform, we're able to more deeply interrogate the immune repertoire and identify single clonotypes that are of interest. So this is the overview of the Trilux antibody discovery process. And I should note that the tri in Trilux actually means a couple of different things. At first, it represents that the platform includes in vivo, in vitro, and in silico discovery technologies, and we use all of them. It also can stand for the fact that we use 3 different hit identification approaches in parallel. And those include Smart Fluidics, which is our single B-cell microfluidics-based platform, XtalPi, which is our NGI plus AI based hit discovery, and then also SpeedPlay, which is our phage display plus AI antibody discovery approach. So in the general workflow, we will immunize an animal of interest. In this case, it was a mouse, and we subject material from those immunized animals to those 3 different platforms. So from the Smart Fluidics single B-cell screen and the SpeedPlay phage display campaign, we're able to identify panels of binders that are validated in the wet lab through primary screening. With XtalPi, we take the NGS data from the immune repertoire, and we subject that NGS data to our pre-trained AI model. And when we do this, we're able to predict a panel of binders. In this case, it was 85. So now what we can do is we can take the sequence information from those 3 platforms and subject it to a second round of AI analysis, which is really an active learning step. So in the first step, we're using a pre-trained model. In the second step, we can refine the AI model using the sequence data from our 3 discovery approaches. And what this allows us to do is identify a panel of binders from that repertoire data that are rare clonotypes. So in this case, we had an additional 52 binders identified to help maximize the overall diversity of the hit panel. From there, we can further apply AI to the workflow using generative AI and also developability predictions to do high throughput in silico humanization. And I'll talk more about that in a minute. But what this allows us to do is go directly from identifying sequences from the campaign, humanizing them, and then expressing and testing the humanized variants in the wet lab. So we don't have to go through the process of testing the mouse antibodies, humanizing them, and then recharacterizing them. So it saves a lot of time and effort in that sense. And so overall, with this particular campaign, we were able to identify 33 functional binders from this Trilux workflow. So to give you a little more insight into our AI engines, I just wanted to share with you, this is the basis for our predictive AI algorithm, and I am not a computational scientist, but this is a high-level overview. We've generated this predictive AI model using multimodal training data. So we have data from multiple sources, including binding data, antibody sequence data, transcriptomics, and also structural features. So those are all taken into consideration with the pre-trained model. And then, as I mentioned in that second round of AI where we perform the active learning step, we're taking sequence data from the binders that we identified, but also using the non-binder sequence data to fine-tune that model and further predict additional binders. So in this particular case, we were able to use AI to catch those rare binders that we don't see in traditional screening. So if you look at the percentage of single clonotypes that appear only once in the NGS data set, once we've applied that second round of active learning, you can see there's a really big increase in the overall number of singleton clonotypes identified compared to just using one round of AI or random selection. So I think this is a nice demonstration of how we're able to really interrogate the repertoire much further than traditional approaches. And to really validate this approach of identifying these singleton clonotypes, we looked at the function of these clones compared to those identified from the other workflows and compared them to a clinical stage benchmark antibody. So what you can see is that using B-cell or phage display approaches, along with our first round of AI using Explore-seq, we are able to identify clones that are on par with that clinical stage benchmark. However, when we do that second round of active learning, what you can see with the clones in orange is we're able to identify a panel of antibodies that show even better function than the clinical stage benchmark antibody. So I think this really demonstrates the value of this particular approach in parallel with our other antibody discovery methods. And then just to comment on the overall sequence diversity in this particular campaign, what I'm showing you here is a phylogenetic tree with the clones from each of the discovery methods. So you can see there's quite a bit of diversity overall, 220 unique H3 sequences identified, and diversity from each approach. So I think that shows the value in taking these parallel approaches on the same repertoire. And in terms of epitope diversity, there were two known benchmark antibody binding epitopes, but we were able to identify three clones that bound three novel epitopes to the target, including this one in green towards the bottom. And that was, we were only able to find an antibody that binds that epitope by identifying those rare clonotypes. So I think this also shows the importance of this particular approach when you're looking for epitope diversity. And as I mentioned, as part of our Trilux workflow, we can do this instant in silico humanization prior to doing any kind of wet lab testing and characterization. So we have developed a high throughput model where we can humanize 500 antibodies in a single day. On average, we're expressing one variant per parental antibody with an 80% success rate. And if you compare the binding affinity of the parental antibody to the humanized variant, which I'm showing you here in this graph, you can see that for the most part, all of the humanized variants are within five-fold affinity of the parental antibody. So we're maintaining binding after humanization. We can also look at the overall humanness score of these in silico humanized variants, and that's what's shown on the right. And you can see that the in silico AI-powered humanized clones are on par with antibodies in the clinic that have been humanized. So they're very similar. And then on the bottom, you can see the overall humanness score of chimeric and mouse antibodies for reference. The other thing that we can do with our predictive AI engine is assess the developability of humanized hits. So as I mentioned, we have this whole suite of predictive models, and we can rank and triage clones before we express them so that we're only expressing hits that are predicted to have good biophysical properties. But for validation purposes, what I'm showing you here is some wet lab data where we took a panel of hits that were predicted to either have good biophysical and developability properties or those that were predicted to behave poorly. And what you can see is overall, looking at these various characteristics, hydrophobicity, thermostability, self-interaction, and polyreactivity, overall there's a high degree of correlation between our in silico predictions and what we're actually measuring in the wet lab. So we're confident that we can use these predictive algorithms to triage hits in a discovery campaign. Okay, so that's hit discovery at Ailux and one example of what we've done there. But I think where AI can really shine is in multi-objective engineering of antibodies. So today what I'm going to show you is how we're able to simultaneously or in parallel optimize features of an antibody, including pH sensitivity, humanization, and overall developability. But before I get into that, really what the starting point for any of these engineering tasks is complex structure prediction using our CrystalFold platform. And so I wanted to share a little bit more information about this platform with you. So as I mentioned previously, CrystalFold allows us to perform antibody antigen complex structure predictions using just primary sequence data. And we have compared CrystalFold head-to-head with AlphaFold 2.3, and that the comparison results are shown here. So what we did was we took a set of structure predictions that were in the PDB database, but were not used to train either CrystalFold or AlphaFold. And we calculated the DocQ scores for those predictions. And if you're in the field, you know that a score of 0.23 is usually the threshold for a good prediction. The higher the DocQ score, the better. And what we see consistently is that CrystalFold outperforms AlphaFold in terms of the DocQ score and the accuracy of its predictions. We have an overall success rate of about 70% compared to AlphaFold 2.3 with 44%. We have not been able to do the same head-to-head comparison with AlphaFold 3 because the code is not publicly available, but our reported success rate is higher as than AlphaFold 3 as well. So we're very confident with CrystalFold, and we've used it on many different projects so far with good success. And CrystalFold has been gaining a lot of interest recently in the field, and we're happy to report that within the last couple of weeks, we entered into a licensing agreement with J&J for their scientists to bring this AI platform in-house to use for their own antibody discovery and engineering efforts. So really excited to be partnering with them there. So getting back to this engineering case study, starting with CrystalFold, that allows us to select candidates that we want to perform engineering tasks on. And then from there, we can perform these engineering tasks in parallel. So we can optimize pH-sensitive binding, perform humanization beyond traditional CDR grafting, what we're calling deep humanization, and I'll share more details about that, and then also performing affinity maturation. And when we identify critical residues important for each of these characteristics, we can then combine them into one final optimized lead. And that's what we have here on the right. So we have an antibody with a fully human scaffold, a minimal number of mouse residues with improved affinity and pH-sensitive binding. So let me walk you through each of these stages. So the first off is candidate selection. Not all antibodies are amenable to antibody engineering in terms of pH sensitivity. And so we like to be upfront about what's possible and what's not. And so part of that is candidate selection. So in this particular case, we took two different antibodies and modeled their binding to their target. And they bound two different distinct epitopes. And in order to validate the CrystalFold prediction, we performed alanine scanning in the wet lab and confirmed which key residues were important for binding. So from there, we can take a closer look at the binding interactions of these two antibodies. So with the first antibody, if you look closely at the binding interface, there are no significant interactions with any negatively charged patches on the antigen. So based on this, it's probably not a good candidate for pH sensitivity engineering. However, our second antibody is interacting with this large negatively charged patch on the antigen that is noted in red there. So based on this, we selected antibody 2 to go forward with further engineering. So in terms of pH sensitivity engineering, this is our typical workflow. Once we have that complex prediction, we look at the key residues that are likely to be able to mediate pH sensitive binding. And in order to reduce the number of variants that we need to search, what we do is an in silico histidine scanning analysis. So we look at these residues and calculate the delta G score. And the bottom line is, the more negative that score is, it indicates that there's a higher chance for pH sensitivity. So based on this in silico histamine scan, we identified 16 residues that are indicated with those white bars that are those that we want to focus on for mutation. So this really narrows down the search space. So from there, we can create a combinatorial yeast display library representing mutations at those residues and perform a yeast campaign and screen for binding either at acidic pH or neutral pH. And in parallel, we can perform what we call deep humanization. And so this is, again, where crystal fold information is really valuable. So in typical CDR grafting, you're just taking the mouse CDRs and grafting them onto a human germline that you think may be most homologous in terms of sequence identity to the parental germline, and then maybe putting in some back mutations. But with crystal fold, we can go beyond just looking at sequence identity and identify germlines that maintain the parental CDR structural conformations. So that may mean they're not necessarily the most similar to the mouse germlines in terms of sequence, but structurally they are more conserved. And so once we do that, we can further narrow down the germline selection. After CDR grafting, we narrow it down by removing J gene diversity, and then we look at the RMSD scores of the CDRs, and then further filter based on predicted developability. So from 29,000 putative designs, we end up with 4 scaffolds that we can move forward with in terms of humanization. And then in parallel with the crystal fold complex structure, we know which residues are critical for binding interactions to the target. And we also know which may affect stability. And then we can identify the non-critical residues within the CDRs. So in this case, we had 27 non-critical residues that we can mutate and play around with. So that's quite a bit of wiggle room that we have there. So we take both of these workflows into account when generating the final humanized variants. And then finally, another optimization task that we can perform is affinity maturation. So in this particular case, we took the parental molecule and performed a yeast display-based affinity maturation approach. And the starting molecule had a low nanomolar 6.4 binding affinity to the target, but after affinity maturation, it was down in the sub-nanomolar range, binding at pH 6. So taken all together, we can achieve these engineering objectives simultaneously with our workflow. So I just want to show you the final results of the optimized molecules. So on the left, we have the parental antibody that's showing equivalent binding at acidic or neutral pH. However, after optimization, we now see that there is quite a big difference in binding between acidic and neutral pH. And then in terms of humanization, we've been able to significantly minimize the immunogenicity risk compared to traditional CDR grafting techniques. So with conventional methods, the molecule would have 73 mouse residues, while with our deep humanization approach, we've been able to reduce that down to 22. When assessing immunogenicity, this deep humanization allows these molecules to really fall within the range of antibodies that are in the clinic or have been approved. And then in terms of overall developability, again, this is something we can take into consideration as we're doing the optimization. We can see that our optimized variant has a much more favorable overall developability profile compared to the parental antibody that's in purple or the conventionally humanized variant. So just to summarize these two cases and what we've learned over the years using AI in antibody discovery. In general, AI is more useful in some cases than others. And there's been a lot of talk about this at this conference this week. And we are not at the point where we can do everything in silico. And not every antibody is amenable to AI-based optimization. And we can find these best use cases empirically, like the case study I just showed you. One antibody was clearly a better choice than the other. And we have found that wet lab and AI capabilities should be combined synergistically to enable and accelerate antibody discovery efforts. And that's why at Ailux we have both of those capabilities working side by side to achieve our clients' and partners' antibody design goals. So today I talked to you about hit generation at Ailux with our Trilux workflow, but we also have a lot of other capabilities that that I'd be happy to share with you. One being that we have a AI-based platform for epitope-specific antibody discovery. We can use AI to design stable variants of your target for epitope focusing. We can also use that similar technology to produce variants of difficult to express proteins like GPCRs or other multipass membrane proteins. We are able to really mine immune libraries through NGS and AI, as I shared with you today. And also, we are anxious and ready to take on your hard targets, like I mentioned, GPCRs, peptide MHC complexes, et cetera. So we're very interested in hearing about your challenges in hit discovery and how we could help you there. In terms of engineering, as I mentioned, we're able to do multi-objective engineering, including affinity modulation, deep humanization, developability optimization, and we can also do species cross-reactivity engineering for your potential leads. So these are just some of the things that we can do at Ailux, and we're always working on developing new technologies and platforms. So with that, I would be happy to take any questions, and please visit our booth downstairs today. Myself and my colleagues will be there, and we're happy to discuss any potential projects, or if you have any other questions or would like more information about our company. So with that, I just want to say thank you for your time today and take any questions.